Welcome back. In this video, I will discuss about the selection process for the isolation of industrially important microbes. The main goal of industries is to convert a simple, low-cost starting materials into a profitable final products. The main player behind this conversion is always a microbes. It can be a bacteria or it can be a fungus. Each and every microbes has some unique features that can be exploited in the industries. Here I will discuss how those microbes are identified and selected from the mixed population such as soil. The main target is to select a procedure to allow easy detection and isolation of only those microorganisms which are of interest from mixed population is called the screening. The main purpose of screening is to allow the easy detection and isolation of the useful microorganisms that are present in the population in very less number. Screening also helps to eliminate many microorganisms that are valueless in terms of commercial application. The first question comes in your mind that from where you should start. We know that microbes are present everywhere, so you can start from soil, water, air, leaves, and different parts of animals. Soil is the preferred source for isolation of microorganisms because it's a vast reservoir of diverse organisms. When proper screening method is applied, it is possible to isolate useful microorganisms from the soil that can utilize virtually any carbon source for growth and metabolism. In recent times, one new habitat called marine environment has been included for screening purpose. New and promising bioactive microbial metabolites can be isolated from marine microorganisms. This process is called biomining. The classical screening method is divided into two stages, the primary screening and the secondary screening. In order to detect and isolate commercially important microbes, the following methods are used routinely. Each and every method has some potential application for the screening a particular type of microbes. Now let's check each and every method in details. The technique is used to detect and isolate those microorganisms that can able to produce antibiotic. At first, 1 gram of soil sample dissolved in 10 ml of sterile distilled water and then mixed and allow the soil particles to settle down. You have to prepare different dilution of the supernatant using the dilution blank. About 0.1 ml of inoculum plated on a sterile nutrient agar plate with a glass spreader. The plates incubated at room temperature for 24 hours to 48 hours. At different time interval, observe the colony which shows the clear surrounding area. Such cultures are then selected, purified and maintained as pure culture. Now let's check the drawback of these method. The appearance of clear zone surrounding a colony does not necessarily select an antibiotic producing microorganism because the inhibition area around the colony sometimes can be due to some other reason like marked change in the pH of the medium due to acid production of the colony, rapid utilization of the critical nutrients in the vicinity of the colony. Thus, further testing is required to confirm the inhibitory activity associated with microorganism is whether attributed to the presence of an antibiotic or not. Another drawback of this method is that which type of antibiotic it is and effective against which type of microorganism. Screening of antibiotic producing microorganism can be improved by these two methods using a test organism and by Wilkins method. In this technique, agar plates which gives a well isolated colonies were flooded with a suspension of the test of microorganism, for example Escherichia coli or Bacillus subtilis. Then the plates are again incubated to allow the growth of the test microorganism. The formation of clear zone indicates that the antibiotic producing ability of the isolate against gram-negative or against gram-positive respectively. A Wilking medium which contains a pH indicator dye called bromothymol blue which is green at neutral pH but it turns colorless at acidic pH. This method is useful for the differentiation of antibiotic producer from the acid producer. Those colonies that produce antibiotic gives a clear zone of inhibition against a sensitive microorganism, without changing the color of the dye. On the other hand, acid producer colony change the color of the dye from green to colorless. This technique is largely employed for the detection of microbes that are able to produce extracellular growth factors. Two plates are required for this technique, one with all vitamin supplement and in other without vitamin. A filter paper strip is put across the bottom of petri dish in this way. The nutrient agar is prepared and poured on the paper disc and allowed to solidify. Soil sample is diluted and proper dilutions are inoculated and incubated. A second plate with a minimal medium lacking all growth factors are prepared and selected with the test organism. The seeded medium is poured onto a fresh plate and the plate is allowed to set. The agar from the first plate is then carefully lifted with the spatula and placed on the second plate without inverting. The growth factors produced by the colonies present on the surface of the first layer of agar can diffuse into the lower layer containing the test organism. The zone of stimulated growth of the test organism around the colonies is an indication that the organism produce extracellular growth factors. This method was first designed by Bayesian to isolate the desired microorganism from a heterogeneous microbial population from soil. This method is useful to isolate very rare organisms from mixed population. For example, if you want to isolate microorganisms that can able to utilize petroleum as a carbon source, they are very rare and low in number compared to other microorganisms. First, the sample is inoculated in a nutrient broth and incubated. Like all other microorganisms, your target microbes will also grow. Now, a small portion of the inoculum is plated onto the solid medium, where petroleum is added as a sole carbon source. After a certain interval, you will observe colonies. Suspected colonies from the plate are subcultured and fresh media and subjected for the further testing. Microorganisms that can degrade starch produce alpha amylase extracellularly. These organisms can be isolated by adding starch into the isolation medium. The diluted soil sample is sprayed onto the petri plate and incubated. After overnight incubation, the plate is flooded with iodine solution. Iodine turns blue where starch is present. Microbes which degrade starch produce clear zone around the colony. This method is used to detect microbes capable of producing organic acids and amines. An indicator dye is added in the medium. Examples of such dye are neutral red, bromothiamol blue, etc. The test organism change the color of the medium according to the pH. So, you have isolated one new microorganism from the soil that may have some potential industrial application. Next, you have to screen further is called the secondary screening. This is accomplished by performing experiments in agar plates or in flask or in bioreactors containing liquid medium. It can be qualitative or quantitative or both. Here are some common characteristics to be evaluated during the secondary screening. The first one, the true potential of the microorganism for industrial usage. That means the product has some public demands or not. The microorganisms are actually producing a new chemical compounds not previously described. If not, don't lose your hope. Try to improve your strain so that it can give a better result. Whether there is a pH or other critical requirements associated with particular microorganism, both for the growth of the organism and for the formation of chemical product. Gross genetic instability of the microbial culture so that it should not change over time. Whether certain medium constituents are missing or possibly toxic for the growth of the microorganism, or its ability to accumulate the fermentation products. In that case, you have to add or remove that particular compounds from the medium respectively. The product has a simple, complex or even macromolecular structure if the informations are not already available. The chemical stability of the product and solubility on various organic solvents. Whether the product possesses physical properties such as UV light absorption or fluorescence or any chemical properties that can be employed to detect the compound during the separation process, like paper chromatography or any other analytical methods, and which also might be of value in predicting the structure of the compounds. If the product have some medical applications, check toxicity against animal and human. Any other byproducts are also synthesized by the microorganism or not. Whether the microorganisms are able to chemically alter or even destroy their own fermentation product or not. So all these questions will be answered when you screen the microorganism for the second time, that is the secondary screening. So that's all for now. If you like this video, subscribe, comment, share and press the like button. Visit the channel community section for more, and finally thanks for watching.